Hello, in this video we will discuss about the mTORC signaling pathway, the mammalian target of rapamycin. The rapamycin is a drug, so that is why it's named through a rapamycin drug. Rapamycin. So the mTORC1 and C2 we will discuss collectively. So this is the cytoplasm inside and the cell membrane, and inside is the nucleus contains genetic material. Remember, you should remember that. Basically it is easy. And the DNA, whenever here is the tyrosine receptor kinase, a tyrosine receptor which does contain extracellular domain and intracellular domain. Extracellular domain, when bind with the growth factor, hormones, cytokines, will trigger the insulin receptor substrate 1 and the PI3K pathway and the RAS, ARC and RSK protein activation for the gene transcription, for several function, for proliferation. So here is the PIP3 will convert into the PIP2 and PIP2 will convert into the PIP3 through PI3K pathway, the phosphoinositide 3 kinase. And in this way, the basically the protein dependent kinase 1 will lead to activate the AKT, and AKT will inhibit the TSC2, TSC1 and TBC1D7 complex. After the inhibition, it will not trigger the RAP. When RAP will not trigger, the TSC2 will basically inhibit. In this way, the uncontrollability will occur for the RAP protein, will trigger the mTORC1 complex. mTORC1 complex contains raptor, daptor, mTOR and G beta L protein. In this way, the adapter will inhibit the mTOR, but how the adapter basically how the rapamycin will work for the inhibition of the mTORC1? It will target to the mTOR, and mTOR protein basically the mammalian target of rapamycin, because it is the rapamycin target. Rapamycin is the drug, remember. So this is the mTORC1 is responsible for the triggering of that genes. Genes contain lipin 1, TFEB, PPAR alpha. PPAR alpha will inhibit, remember, and this inhibition, while the HIF1, PGC, the PGC 1 alpha and the PPAR gamma, SRAB-D1 and SCAR. This protein is basically responsible for the ribosome. Lipin 1 is used for the ribosome synthesis. TFEB is used for the autophagy, lysosome biogenesis. While the PPAR alpha will inhibit, and in this way the lipid metabolism will inhibit. While the HIF1 gene is used for the vascular endothelial growth factor, is used for the angiogenesis, the blood formation, blood vessel formation, for the tumor formation. And in this way, the PGC1 alpha will lead to mitochondrial metabolism, and PPAR alpha gamma is used for the adipogenesis and the lipogenesis to SRAB-D1. This all gene is triggered via a mTORC1, which that activate the genes to transcribe, and this transcription to translate that protein for the survival of the cell as well as proliferation. While this is the taurine 1, PP242, Q 63794 and WYE354 is the gene will inhibit this mTORC1 and 2. Let's begin to understand the mTORC2. mTORC2 complex, what is this and how it differ regard to the mTORC1? This contain a SYN1, PRR5, protein and Richter and mTOR and GB ligand and daptor. This daptor will inhibit, and this is basically it will trigger the mTORC2 will trigger to synthesis glycogen kinase 1 and the protein kinase C alpha. While here is another protein, the 4E binding protein 1 and 2 and messenger RNA transcription will inhibit, and in this way the proliferation will inhibit. while this will be inhibited through mTORC1. After this inhibition, the proliferation will trigger. So remember, this is the another concept for understanding. Let's begin to understand the mTORC2 pathway. It is same mechanism, and the growth factor, hormone and cytokines will trigger this, but I am drawing the separately. So in this way, the synthesis of glycogen kinase 1 and protein kinase alpha will stimulate. In this way, the proliferation will maintain and the cancer will progress. Anyhow, let's begin to understand the further process. In this way, the mTORC2 will activate the AKT. It will activate the AKT. After the activation, will AKT will lead to again inhibit, and in this way, the RAB will activate. Let's further understand the DNA damage. When the DNA damage should be trigger the p53 gene for the cancer inhibition. In this way, when the p53 gene will trigger, it will lead to cysteine 1 and 2 gene activation, will lead to trigger the AMPK pathway. This when AMPK pathway will trigger, will inhibit the mTORC1 complex, will lead to trigger autophagy automatically. But whenever the DNA will damage, the p53 gene will not controlled, and in this way the p53 gene will inhibit due to the DNA damage and mutation. This will lead to not trigger the p53 genes. In this way, the inhibition of the mTORC1 will not inhibit. While on the other hand, the mutation will trigger the RAS and mice gene uncontrolled transcription, will lead to that protein production will inhibition to cause cancer of the mTORC1 triggering due to the inhibition of the AMPK pathway. So that is why the cancer can cause due to the DNA mutation. So anyhow, further on we will also learn about the hypoxia. The stress will also trigger the red 1 and 2 genes, and here is the vent pathway, which that is bound with the receptor and LRP domain, and the G couple protein alpha Q and 0 and DVL will inhibit the glycogen synthase kinase 3. After this, glycogen synthase kinase 3 will inhibit. In this way, the TSC2 will not trigger, and in this way, the inhibition of the RAB normally. And hypoxia also will lead to trigger the RAD1 and 2 will trigger the TSC2 will inhibit the RAB, and in this way the RAB when inhibit, in this way it will not mTORC1 triggering mechanism. While the glucose and AMP and ATP ACOR is also trigger the AMPK, will lead to inhibit the mTORC normally, but if will not control, lead to cause cancer. Glucose amino acid also trigger the mTOR 1, 2, 3, 4, 5. This mTOR will trigger the RAG A and B as well as RAG C and D. And in this way, the GTPase enzyme will convert, and GDP will activate, and in this way, the activation to mTORC1 translocation to lysosome. And in this way, the mTORC complex will translocate to the lysosome, will lead to inhibit the lysosome activity, and in this way, the proliferation will maintain. While here is the metformin also inhibit the RAG A and B, and when inhibit the mTORC1 translocation to lysosome will inhibit. In this way, the metformin is very important for the inhibition of the cancer. So here is the mTORC2 will trigger mTORC1 translocation to lysosome will also trigger the raptor to working further on. And here is the GATR2 complex, which that's contain mice VDR24, VDR59, and SAC13 and SEH1L will trigger the GATR2 complex, another GATR1 complex, will inhibit the RAG A and B. So anyhow, when a DNA damaged cell should be apoptosed to prevent cancer. If these events not occur, cell progress to replicate and cause cancer. So the simple for understanding throughout the video, the simple is summary is that when DNA damage, cell should be apoptosed. If not, it will lead to proliferate, will lead to uncontrolled cell division due to the mutation will lead to cause cancer. So I hope you make sense about these. Please make sure to subscribe, like and share. Thanks for watching. Bye.